Karim, the floor is yours.
Yeah, thanks a lot again, John Glass, for letting me present here. My background is actually in infrared spectroscopy and fluorescence spectroscopy of membrane proteins, and more recently, we started also doing so-called heat flow measurements from entire cells in the context of addressing toxicities of metals. And well, whenever you do lipid research in Dresden, of course, you know James Science, and I'm very happy that we could team up to actually use these heat flow experiments that we had set up in combination with minimal cells. Great. So isothermal microcalorimetry, that's what I'm going to talk about in the context of systems biology. That's an interesting and technically well-developed field, but theoretically it's still underexplored. It's a method that captures essentially all metabolic processes in a cell by measuring the heat that is produced by metabolism. And that's essentially, so to say, some signal that collects all the delta H's of cells' metabolism. And it is conceptually very easy. So you have a ampoule which you fill with the cells in a medium. And these are typically 2 milliliters only. We put that into a 4 milliliter ampoule, and that's put then into contact with a heat sink and an apparatus which stabilizes the temperature. And what we measure is not the enthalpy as such, but the heat flow. That means the temporal change of the sum of all delta H's that are generated as exothermic reactions by the cell. And such a heat flow curve would then essentially look like this. It's a power, really in microwatts, measured over time. And when I started doing these experiments, I realized that there's no mathematical framework at hand that would allow us to analyze these data in more detail. So we live essentially only with a peak and a time, but we try to do more. And the interest in using minimal cells is two-sided. It's, of course, we want to understand the metabolism of these cells, but also from the perspective of heat flow analysis, it may help to benchmark the mathematical approaches that we have chosen to analyze such data. We try to do this in the context of the simplest. We try to do this in the context of the Monod equation, which is plotted here. That's describing hyperbolic dependence of the metabolic activity of a single cell with respect to available substrate. What I prefer is to present it in this way. The reason is simple. We want to apply this equation not only to the exponential phase as originally defined, but to the whole metabolic lifespan of a batch culture of minimal cells. And these cells, of course, start with a high substrate content, a few cells, and they end up with a low substrate and many cells. That's why I would say, looking at the Monod equation, that mirror imaged way aligns the data, so to say, with respect to an implicit time constant. So the batch culture goes from high to low, and in parallel, it goes from low to high in terms of biomass. And what we do to describe minimal cells batch culture heat evolution is we multiply these curves for a simple reason. In the beginning, you have lots of nutrients, but little biomass. So the heat flow from a batch culture should be just the product of both. Somewhere you have a decent nutrient amount and still decent metabolic activity and a reasonable amount of biomass, so you get a good signal. At the end of a culture growth, you again run into a zero signal because all the nutrients are consumed. Although you have lots of biomass, there's no heat flow because the cells have run out of nutrient. The reason I explain this is to make you familiar with the way we represent such data. This multiplication yields an inverted parabola shape if we plot the heat flow of a batch culture versus the heat that it has released. And as we know from the yield equations, the heat release is, of course, proportional to the biomass that has accumulated because metabolism and growth are tightly coupled in these cells. So in other words, we will discuss these parabola-shaped curves which we describe, and you can rediscover here the Monod equation in that right part. We just have replaced substrates by joules, by enthalpy terms, and the Monod constant is converted into a parameter, just a fitting parameter that corresponds to the heat that a culture would have released if it had started growing at half maximal metabolic activity. So that is all the mathematical background. Here you see the multiplication that I've just explained with the biomass. And again, heat or enthalpy is a proxy of the biomass of the system. The problem, the advantage of using this presentation is that here in the enthalpy domain, we can formulate analytical functions, which we cannot do in the time domain. But we can numerically transform back into the time domain and then really simulate our raw data. And that's what I'm showing you here with Syn3B cells, where we have the raw data, the heat flow data of Syn3B cells grown in a minimal diet that James had studied already and defined. That's 2 fatty acids only, plus cholesterol. And you see the cells grow nicely over time. They reach a peak as expected, and they decline again. And a large part of that heat flow is really totally consistent with this extended Monod model, which now goes far beyond the exponential growth phase. And the heat that these cultures release is the integral under the curve. So this is really power, heat flow in microwatts, that is time, and watts times time multiplied with time gives the energy. And in the enthalpy domain, this orange area here translates in the length of this arrow. And that is a very important number because it tells us how much energy the batch culture would release if it had continued growing in the exact Monod type of growth that we use to model the system. Even though you see the cells start deviating at some point, probably due to acidification of the medium or other effects that I cannot discuss here. But the important message is we get a number that is in joule that represents the amount of heat the cells as a batch culture would release if all their nutrients would have been consumed. And of course, naturally, the outcome is also the maximal rate of the cell division. And if we do the same thing with a diet that additionally contains sphingomyelin, we were really surprised to see that the peak is lower and the curve is narrower, and we have a much larger effect than I could hope for, because it's obvious that the total released heat is much smaller for a sphingomyelin containing diet than for the only 2 fatty acids diet. Now, if we look at these factors, there's more than 50% reduction in the heat release, but there's not much change in the maximum rate of the cells in their division. So these are phenotypic differences, and we have done this, of course, with the purpose to assign contributions. So why is one culture producing more heat than the other in a response to lipid composition? So our idea was there must be fractions that go into biomass and other fractions that of the nutrients that go into other processes. And a way to use calorimetry to find out how energy flows are separated into biomass forming activities and other processes is only by using a reference diet. We cannot do this in absolute terms. We can only say in comparison to, for example, the sphingomyelin containing diet, the 2 fatty acids containing diet has such and such percentage of nutrient feeding into biomass production and such percent into non-biomass production. And again, without going into details, this K can be determined from our calorimetric data from these Monod type of fitting constants, HF for the reference and HF for the sample. Now, if we do this, we can look at these 2 data here in the bar graph. This is the total amount of heat released by the reference diet. That's the total amount of heat released by our 2 fatty acids diet. And you may think, okay, the difference must be lipid related. But that cannot be true because they've grown in the same amount of nutrient. So how can I have the same amount of biomass formed if I have, with the same amount of nutrient, produced more heat? That means by definition, the biomass must be less because more heat has been dissipated and less energy from the nutrients would be available for biomass formation. And if we use that criterion that I've just shown based on these monotype constants, in fact, what we predict from the heat flow curves is that with 2 fatty acids only, less than half the biomass should be formed in that diet as compared to our reference diet where we have additional sphingomyelin. Now, this is an interesting heuristic approach. There's no independent proof for this, but that's a heuristic approach, and it is hopefully justified because the metabolism is simple. Now, we did this for many diets, for 7 diets, same dissection into biomass forming heat related dissipation and in non-biomass forming reactions. And we see very systematically that if we sort all our cultures according to the degree of total heat, which is the total bar height here, then I have already sorted them in the order of increasing biomass. So it seems to be very convincing. And since the rates that we observed, also from the microcalorimetry evaluations, do not show this monotonic behavior, but are more similar, we thought that seems to be an indication that if I have less biomass, for example, here in the 2FA's diet than in the reference, but still have relatively comparable division rates, that seems to suggest that the cells become smaller in the diet that produces less biomass. And actually, we studied this by DLS, differential light scattering, and it was really a blind double-blind study, so to say. Natasha Safronova did this experiment then as PhD student in James' lab, and I just told her, here, do these size estimations and sort your data in the following sequence. And that is what came out. A wonderful correlation between the predicted biomass, dark red, and the size of the cells. If we have the size, the diameter, we have, of course, also the volume. And if I put the volume here, you see that the yellow part and the dark red part are always in very good proportion to each other. That makes me believe that this heuristic mono-based approach is really biologically meaningful. Now, what the outcome is that I think that the lipidome size affects the cell volume by retuning the balance between cell division and biomass formation. So a minimal diet produces less biomass, not because there's less cells being formed. There's relatively many cells, but they're just smaller. So it's interesting. Lipidome size really affects cell volume. If we want to compare this eventually with lipidome, we have more things to do. First of all, we thought it could be that the altered lipid composition affects negatively the nutrient uptake. And that is also information that is encoded exclusively in the heat flow curves. I don't go through the mathematics, but this this bluish greenish bar here shows the nutrient flux per surface area, which we have here determined from the DLS data. And if we compare this with the reference diet, the 2 fatty acids plus sphingomyelin, we see that all the other lipid diets have rather more nutrient flux than less. And that means that we cannot explain the decrease in biomass formation by lipidome reduction. So it seems that the import of nutrients is not impaired by the different lipid diets. Now, if we compare this with lipidomic analyses that James had performed previously, you see that the situation is complicated. We have so many numbers in the upper panel and many numbers in the lower panel. It's hard to compare this, but one way to do so is to define a culture growth performance parameter as a number value, which is again taken from calorimetry. It's just the rate, the division rate, the maximal division rate, multiplied with the predicted biomass in form of its heat equivalent. And if we do so, we see that this growth performance seems to be high whenever the lipidome is diverse, and it goes down when it is less diverse. And if we want to assign specific functions to specific lipid classes, roughly, one would say DO, the presence of DOPC, that's abbreviated as PC here, seems to increase the rate, the green bar here, whereas the presence of sphingomyelin seems to increase biomass. So lipid diversity is key to growth performance. Sphingomyelin is good for biomass, DOPC good for division rate. But how does it correlate now, or how can it be explained with other lipid properties? And if we focus on these two comparisons here, 2 fatty acids and 4 fatty acids, still no sphingomyelin. Already 4 fatty acids gives a big advantage in biomass and growth performance. And if we look at the chemistry, however, if I form phosphatidylglycerol, which is the predominant here in the 4 fatty acids in the better growing culture, it would pick out one C3 metabolite from glycolysis to put it to 2 lipid acyl chains. Cardiolipin, for example, which is more dominant in the 2FAs, would be picking out only one C3 metabolite for 4 lipid side chains. So I think from a chemical point of view, it's more efficient to do cardiolipin than phosphoglycerol, but nevertheless, the more resource demanding lipid still enhances the growth. So I think that lipid chemistry will not explain these large delta H differences. And there's other arguments that speak against that. So it seems to be that it's maybe global lipid bilayer properties rather than lipid chemistry that affects growth performance. And of course, you all know the different curvature tendencies in these lipids. And one could think of during the cell division, you need certain curvatures at different sides of the cell. So it may be good to have cylindrical lipids in some cases, or negatively curved lipids like cardiolipin when the cell is dividing. So that's one thing that could maybe explain through mechanical properties the large amount of heat dissipation, kind of an inefficient way of doing the cell division. That's one aspect, the mechanical work. However, another could also be that it still has to do with membrane lipid, with lipid protein interactions. And if we look at the ZF, for example, that is going to bind to the lipid membrane, then of course also ionic interactions play a role. And I think that may be a cause why the lipid composition will also affect these electrostatic interactions. And my conclusion here is that heat flow experiments show that probably it's the divisome that is affected by the different lipid composition, and that has a tremendous influence on cell metabolism, and it's not lipid chemistry that is affected so much. And with that, I'd like to conclude. So I think it's non-productive lipid-protein interactions in the divisome that we measure by heat flow, and it would be an outlook also that in the future one may use these isothermal calorimetric data to study in more detail using mutants how the machinery of the cell division is linked to energetics of metabolism. Yeah, thanks a lot.
Wow, I'm dazzled, Karim. I really am. This is a new technology from at least our organization's perspective, and it really has me thinking about a lot of things. Have you considered trying doing your microcalorimetry with JCVI Sen 3.0, which does not have FTSZ, will not make a Z-ring?
Yeah, as I told you, this is a very fresh data and a young collaboration. So I think there's many ideas. Also with James, I've discussed many further options. That's certainly one, because also from the previous work that I did with calorimetry, it is as such not a very informative technique. You have to combine it with genetics or with very well tailored and manipulatable organisms. Otherwise, it wouldn't tell you much because it's just a sum signal. And to make it assignable to certain processes, exactly that's what you said is the right way to go. So yeah, it's a huge field, I think. And these are just the first data. We do have mycoidids data as a reference, and we have also full diets. And we start paving our way through these data to come up with new ideas and new results.
One experimental suggestion at the 6 a.m. talk, Malik Ali from the Chan Zuckerberg Imaging Institute is going to talk about doing cryotomography of minimal cell. But you might work with her and send her some of your different media to get volume measurements for cells grown under different conditions, possibly. And that it would seem that she has capacity to measure volume more precisely than anyone else.
Oh, that's a good suggestion. Right. Yeah, DLS is tricky, and it has variations, and also cell numbers. I didn't talk about cell numbers. We have done cell number estimates, and I just want to mention, roughly, we end up in the femtowatt range of per cell heat flow. That is about 1 per mil, or 10 or 1%, less than 1% of what we typically measure with E. coli, for example. So we can pull many more numbers out of that assay if we combine it reasonably with other data. But I didn't mention cell numbers in absolute terms because it's also very, very difficult to determine them accurately.
There is a new technology that I'm getting ready to buy for our lab. As opposed to doing conventional cell number measurement methods like colony forming units or color change units, this instrument called BactoBox, I think it's a Danish company, but they do a sort of for $5,000 you get a device that will give you in 2 minutes a fairly accurate measurement of the number of cells in a sample. I'll put information about this in the chat. So Prescott Atkinson at UAB sent me some information. They presented at the recent International Organization for Mycoplasmology Congress. So it's something you might consider. Maybe next year or later this year we and others may show results, but this might be a technology that could be useful for what you need. Other questions for Karim?
Karim, James Dobbins Peck in Birmingham, Alabama. I was just curious, did you see any difference in the osmotic strength of the bacterial cell when you changed the lipid diet?
That's a question to James. I think that's rather a question to James Science, right?
Yeah, so we've done sort of a parallel analyses on the same diets, and yeah, we did osmotic sensitivity analysis, and there are differences. I have to pull the figure up, and we have a manuscript ready with that data in it. But yeah, there are definitely distinct differences, and my recollection is that they became less sensitive to hypoosmotic shock when we gave them things like external lipids like PC and sphingolipids.
That's what I would have expected. Thank you.
Sure.
Related to James Dobbins Peck's question, James Pelletier here, beautiful and inspiring work. I know that you've also looked at permeability of the membrane with the fluorescein diacetate assay and dissipation of a transmembrane electrochemical gradient. I wonder how that could contribute to the futile heat dissipated. And just, I'm sure you're already thinking of it, but correlating the efficiency of growth with the amount of protons dissipated without providing a cofactor for transport of metabolites. I'd be really curious about that as well.
So for me, Karim, yeah.
Yeah, I would say that's again a beautiful suggestion. I mean, as I said, these experiments take fairly long, and also the evaluation takes quite long. They grow slowly and so forth. But these are exactly the lines along which one should think. So what I try to convince you of is that I think that calorimetry could be a wonderful additional learn understanding these they grow and many directions.
Other questions?
Yeah, this is Kim Wise. Karim, this is fabulous work. I've been hearing from James that this is going to be happening. I got a question, though, about the difference, the homeoviscous remodeling that was talked about earlier in another session. As I recall, things that do not require synthesis, that is like sphingomyelin, are contributing to the membrane structure, whereas most of it, as I recall, the homeoviscous, the adaptation is dependent on things like PG and cardiolipin and the things that are synthesized. So I wonder if there's a relationship, if what the energy difference is to get something like a sphingomyelin in, I'll say passively. I just mean without ATP and synthesis. So can you make a case or even test this with your technology to see if there's a difference in those energies?
Yeah, this is a very interesting question. I think that we can only address also in combination with explicit models, because we had an interesting discussion with Zan Luthi Schulten, because my hope was that we can combine these experiments with the predicted enthalpy changes from the chemical or metabolic network that she has set up. So in essence, she has all the data to predict the enthalpy that should be released when I build one cell. And recently she said that she has now also included lipid synthesis, which was not included in the previous models. So my hope is that we come exactly back to that question once we can compare it with predictions. I don't think we can pull out that information purely from calorimetry. I think, again, it needs to be contrasted by model calculations. But my general feeling is that lipid chemistry is not telling the story. It's such a large difference. I think it's more than that. And what is often forgotten in discussing these lipid diversity is when we talk about carbolipin, which would be sorted into negative curvature regions. That's well described during cell division. Then what we do there, it's not only curvature contrast, it's also an electrostatic contrast that we produce, because we pull also negative charge there. And if we add neutral lipids or net neutral lipids like DOPC, it could be that those just help to increase the contrast between the charge contrast between the negatively curved region and a positively curved region. And as long as we don't have this mixture also not only of curvatures but also of net charges, it may be, that's my hypothesis, it may be that the divisome, the ZF, for example, would not know where to attach to the memory because there's not a clear preference given by curvature. It may, so to say, diffuse around without ever forming a narrow ring-like structure, whereas in the presence of neutral lipids, it would, because then the curvature would also define the charge distribution. And that's why I think about membrane amphipathic interactions, cytosolic proteins interacting with charged membrane surfaces being more important. And so to say, whatever uses ATP would run idle as long as the right structure has not formed. And that, so to say, is what lacks then in the path for biomass formation. So my feeling is that these are not really separated processes. A part of biomass formation should go on, although the cell already starts to divide. And then the division machinery runs idle because it doesn't find the right structural organization, and the biomass suffers because it doesn't have the ATP to do synthesis. Something like this.
One other real quick question. So what else do you need for your data to give you directly the biomass? If you have a volume, can you measure biomass directly? Because that's been a real problem to do biochemically.
Yeah, as John said, I mean, if there's a good machine that could give us cell numbers and another machine that gives us cell volumes, we would be happy. But at the moment, we have to guess. And I'm happy enough that the calorimetry seems to predict at least the right tendency. It's very consistent. We didn't have to tweak anything in the data. It's just an outcome in parallel.